Hello, and welcome to the Clinical Care Options Oncology Podcast. I'm your host, Tim Quill. Today's episode features Dr. Alexandra Leary from Gustave Roussy Cancer Center in Paris, France, and Dr. Domenica Lorusso from the Fondazione Policlinico Gemelli in Rome, Italy. They will be discussing key updates and new clinical trial data for cervical, ovarian, and endometrial cancers that were presented at the 2023 European Society of Gynecological Oncology Annual Congress held in Istanbul, Turkey. This episode is part of a larger educational program entitled Key Findings and Expert Recommendations in Gynecologic Malignancies, Independent 2023 Conference Congress coverage. For more information on Dr. Leary and Dr. Lorusso, along with a link to the complete program, including a downloadable slide set and clinical commentaries, please visit the show notes for this episode. Now let's get started and hear what the experts have to say about key data presented at the 2023 ESGO Annual Congress.
Hello, my name is Dr. Alexandra Leary. I'm a medical oncologist at Gustave Roussy Cancer Center in Paris, France. And I'm pleased to be co-hosting this with my very distinguished colleague, Dr. Domenica Lorusso, who is a professor in the Department of Gynecological Oncology at the Fondazione Policlinico Gemelli in Rome, Italy. Welcome, Dr. Lorusso.
Oh, thank you, Alessandra. It is a great pleasure for me to be here with you to discuss this interesting ESGO meeting.
Today, we will be highlighting for you data from some of the key studies that brought to light new data at the 2023 ESGO Congress that was held in Istanbul, Turkey. We will share with you our views, our interpretations, our key takeaway messages on the data that was presented live regarding endometrial, ovarian, and cervical cancers. The first study I would like to present, and my colleague Dr. Lorusso will be speaking about the long-term follow-up from the SIENDO study evaluating Selinexor, which is a nuclear export transporter inhibitor, compared with placebo as maintenance in patients with p53 wild-type advanced or recurrent endometrial cancer. In addition, we will be presenting data again in endometrial cancer regarding a phase 1/2 study of a novel ADC against HER2 and recurrent metastatic endometrial cancer expressing HER2. Finally, we'll be presenting the primary analysis from the ICON-8B study, evaluating weekly dose-dense chemotherapy plus Bevacizumab compared with every 3-week chemotherapy and Bev as first-line treatment in high-risk epithelial ovarian cancer. So first, Dr. Lorusso, can you tell us a little bit about a major new key results in a study in endometrial cancer, in particular the SIENDO trial?
Oh yes, the first study that I want to comment with you is the SIENDO trial. As you remember, SIENDO trial is the first trial evaluating Selinexor, exportin inhibitor, exportin 1 transporter inhibitor in the maintenance setting of advanced or recurrent endometrial cancer in comparison to placebo. As you remember, at the first presentation of data, when we reviewed the research the first time in this population of stage 4 patient in 44% or recurrent patient in 53% of them, the results were not so good. In the overall population, the trial did not meet its primary endpoint, but very, very interesting. According to the mechanism of action of Selinexor, which is an exportin 1 inhibitor, it seems to be a great rationale in exploring different effects of Selinexor in p53 wild type or mute patient. This is exactly what was presented at ESGO. The maintenance treatment with the Selinexor in the p53 wild type population translate in, and I have to say, impressive increase in progression-free survival from 5 to 27 months. We did not have the same figure in the p53 mute population, where we did not see any benefit and potentially also a detrimental effect. But we can have some overlapping population in endometrial cancer, and some P53 wild type patients present microsatellite instability, some other no. When we try to evaluate this subgroup of efficacy according to the microsatellite status, what was evident is that in the TP50 wild type pMMR population, the benefit was really high, from the median PFS increase from less than 5 months to a not reached in the Selinexor arm. We have also good results in the MSI high population, with a median PFS increasing from 3.7 months to 13 months. So the conclusion is that the benefit was reported regardless MSI high status. What is interesting is the toxicity profile of the drug. Nausea, vomiting, and diarrhea and constipation, so gastrointestinal toxicity, are the most frequently reported toxicity, but likely in most parts of patients were grade 1, 2, and grade 3, 4 was only diarrhea in 4% of patients. There is also some hematological toxicity, with the thrombocytopenia reported in 42% of patients, 9% of them grade 1, 2, and neutropenia, 34% of patients, 20% grade 1, 2. But in general, the toxicity profile was quite good.
Thank you. Thank you for that excellent summary, Dr. Lorusso. The other key study in endometrial cancer we wanted to mention today is the open label phase 1/2A study of the HER2 targeting ADC called DB1303 in recurrent or metastatic endometrial cancer. Here I'll remind you that there was great interest at ASCO with a similar study, a different compound, but an ADC targeting HER2 in HER2 positive endometrial cancer with beautiful results. Okay, so here we have a second agent. This study recruited women with endometrial cancer previously pretreated who expressed HER2, and any level sufficed, 1+, 2+, 3+, or positive for amplification. And here, this was a single-arm study where patients were treated with the ADC. It included a total of 32 patients. Importantly, 60% of the patients already had had an immune therapy, and 35% already had had a HER2 therapy. And here we see really interesting activity with an overall response rate close to 60% in the highest dose group. I mean, really interesting response rates in patients, whether they were 1+, 2+, 3+, and it included even carcinosarcomas. So really encouraging activity, overall quite well tolerated, with only 30% grade 3 toxicities, mainly anemia and hypokalemia. So really very, very encouraging data for this new ADC targeting HER2. And again, regardless of the level of HER2 expression. So this includes HER2 low endometrial cancer.
During ESGO, results of another subgroup analysis of KEYNOTE-826 were presented, the subgroup analysis of the efficacy of pembro according to the Bevacizumab. You will remember the trial design. This is a trial evaluating the combination of platinum paclitaxel plus or minus Bevacizumab plus or minus pembrolizumab in a population of advanced, recurrent, or metastatic cervical cancer. Bevacizumab was given according to the physician preference, was not mandatory for all the patients. So in the trial, most patients, 76% of them, receive Bev, but when we try to understand what were the reasons why Bev was not given, most frequent reasons were medical reasons, the risk of gastrointestinal fistula, hemorrhagia, and the worsening of hypertension. But in some situations, the drug was not approved in the country or was not considered standard of care. In terms of patient characteristics, even though this was not a stratification criteria, but nevertheless, the two groups appear quite well balanced with a slight numerical imbalance. In general, PD-L1 negative was reported in about 10% of patients. 70% of patients have received prior radiation alone or in combination with chemo. 30% of patients were stage 4B at diagnosis, and about 20% of patients came into the trial with a metastatic disease. Looking at the final overall survival, a significant increase was reported of pembro in combination with chemo, regardless the use of not of bevacizumab. But looking at the other ratio in patients receiving also Bev was 0.56 with respect to 0.61 in patients who did not receive Bev. And this was true and confirmed also in the all-comer population, not only in the PD-L1 positive patients. On the same page, overall survival benefit was reported regardless the use of bevacizumab, but for sure the benefit when we combine pembro plus bevacizumab seems to be very impressive, but also in patients without bevacizumab, a very similar other ratio, 0.60, 0.61 was reported. Again, this was confirmed also in the overall population. Secondary endpoint was the duration of response and the overall response rate. When we combine a significant increase in overall response rate and duration of response was reported in patients receiving pembrolizumab, regardless we had or not bevacizumab, but for sure, when we look at the 77% response rate achieved by patients receiving Bev plus pembro plus chemo, 30% of them were complete response. These data are really impressive and never reported before in cervical cancer. The duration of treatment was longer in the pembro arm, so this at least in part can justify a 10% increase of grade 3 adverse events and also treatment-related adverse events, a slight increase in serious adverse events, and in events leading to dose discontinuation. Of note, no difference in events leading to death was reported. In general, the most frequently reported toxicity were alopecia, anemia, and nausea. The addition of pembro does not increase in a significant way the toxicity with respect to patient does not receiving pembro, and as expected, the patient receiving bevacizumab experience higher risk of hypertension and proteinuria. But of note, when we look at the toxicity of interest with Bev, I mean perforation, hypertension, and fistula, and embolism, the addition of pembro does not increase the toxicity. In an exploratory analysis, the authors try to address what happens when you discontinue bevacizumab for any kind of toxicity. Looking at these curves, pembro continues to work. And in patients who discontinue Bev for any reason, mainly toxicity, you have to continue pembrolizumab because the patient still continue gaining benefit. I think that these data are really practice changing, and that this combo is the new standard of care in the first-line setting.
Thank you very much, Dr. Lorusso, for that marvelous summary. It's fascinating to see that the benefit is really across all subgroups. Now I'd like to move on to talk about key data presented on ovarian cancer. Dr. Lorusso, can you get us started with the really highly anticipated results from the ICON 8B study? And then I'll wrap things up at the end with our study on HRD testing on cell-free tumor DNA from peritoneal fluid. Thank you.
Very good. During ESGO, we saw another very interesting study, which is the ICON 8B study. I want to remember you that at the first trial design was a randomized phase 3 trial comparing carboplatin paclitaxel bevacizumab standard every 3 weeks versus weekly platinum paclitaxel without Bev versus weekly platinum paclitaxel plus Bev. The arm B, the weekly without Bev, was discontinued because of futility. So the trial moved further as a clear comparison between every 3-week chemotherapy plus Bev or dose-dense weekly chemotherapy plus Bev in a population of newly diagnosed high-risk epithelial and ovarian cancer, stage 3 with residual disease, or stage 3 requiring neoadjuvant chemotherapy, or stage 4. I mean, a very high-risk population. And in fact, the patient characteristics are really support the high-risk population. 40% of patients were stage 4, 50% of patients were stage 3 with residual tumor. Primary surgery was received only by less than 15% of patients, and delayed surgery after neoadjuvant chemotherapy was the main treatment for most of patients, 85% of them. So we are talking about really an high-risk population. The treatment were very well tolerated. It's not surprising to see that this trial confirmed the less toxicity of the every 3-week administration. Look at the patient that completed the treatment, which were 67% in the arm B1 and 53% in the B2. And looking at the toxicity, hematological toxicity, look at anemia was higher in the arm B. Look at peripheral neuropathy was similar. So the conclusion is that every 3-week and weekly paclitaxel does not create a problem. Hypertension seems to be a little bit higher in the arm 1. This is quite difficult to explain because both the patient receive Bevacizumab. But most of our patients, as I told you, delayed the surgery. And for this high-risk patient, it seems that every 3-week treatment work with respect to the weekly treatment. Let's move to the results. Surprisingly, because we had several negative trials in the Caucasian population exploring the dose death, but surprisingly, ICON-8B reported a significant increase in median progression-free survival in patients receiving the dose-dense weekly regimen, with the median progression-free survival increasing from 16.7 to 22 months, which is really impressive. And also an increase in overall survival was reported for the dose-death treatment. Looking at the forest plot, there is a consistency of benefit basically in all patients. And when we look at the benefit with respect to the surgical approach, the benefit, as I anticipated, seems to be higher in patients who have received delayed surgery. So I mean in patients coming from the dose-dense treatment after neoadjuvant chemotherapy. In patients who receive primary surgery, there is also a significant benefit, but not so huge as in patients receiving delayed surgery. As I anticipate, also in terms of median overall survival, we saw a clear signal, and median overall survival increased from 41 months to 51 months in arm B1 and B3 respectively.
Dr. Lorusso, thank you for this great summary of the ICON-8B study. So data gets confusing. We get one study in Japan that says weekly is better than 3 weekly. Then we get European study that says there's no different. And here we're back again that weekly is better than 3 weekly if you put it with Bevacizumab. Okay. How do you interpret? What should we do with this data? Should it change our practice tomorrow or not?
Oh, Alexandra, it's very difficult to change a standard of care which is in place since more than 20 years with only one new trial reporting data which are, as you say, inconsistent with respect to what we had until now. We tried to explore several times the strategy of those dens, and we always failed in the Caucasian population. How can we explain this different data? In my opinion, this is a completely different population. We are talking about a very high-risk patient. Most of these patients receive neoadjuvant chemotherapy with interval debulking surgery, 40% of them were stage 4. So all stage 3 should have residual tumor more than 1 centimeter. So probably this is a very high risk population. My interpretation, but it is only an hypothesis, is that we know that weekly paclitaxel, dose-dense paclitaxel, may have also an anti-angiogenic agent. So potentially, when you have a huge amount of disease, you have much more vascularization. And in this respect, probably the weekly administration and the dose-dense administration may play a role. But honestly, to say this is a new standard of care, I would say not. I would say not also because the standard of care now it's represented by also PARP inhibitor in some situation in combination with Bev or PARP inhibitor alone. And this trial, which started several years ago, does not include what we consider actually the new standard. It's absolutely an alternative can be considered in high-risk patients, taking in consideration the different toxicity profile that, particularly in terms of anemia, is worse than for the patient receiving the dose-dense scheme.
Okay, now I'll just finish this session with HRD testing on cell-free tumor DNA from peritoneal fluid of patients with newly diagnosed epithelial ovarian cancer. Now here in this study, we basically wanted to see if we could use a different kind of sample to do the HRD testing. We know we need to do it in everyone at diagnosis. We know we have problems with doing BRCA mutation and genomic instability testing on FFPE tumor samples. Problems we have is that samples are small. We sometimes have non-contributive test results. 15% of our patients, we don't have an HRD status on them. And sometimes the DNA is simply not good enough quality, so we risk missing large BRCA rearrangements. We know cell-free DNA from blood doesn't work very well in ovarian cancer. But what's another source for liquid biopsy in ovarian cancer is peritoneal fluid. Okay, we had done a preliminary feasibility study that showed that with just 20 mils of peritoneal fluid, we could do NGS, get great quality cell-free DNA. And so we then aimed to prospectively validate this. Okay, and so we took women with newly diagnosed ovarian cancer who had a diagnostic laparoscopy, or women who had maybe been diagnosed elsewhere and came to us after 3 cycles of chemotherapy and had a diagnostic laparoscopy again. And we were able to collect at least 20 mils of peritoneal fluid in the vast majority of these patients and in 100% of patients at diagnosis. In 98% of cases, we could get cell-free DNA, which we could confirm was tumoral because we found a p53 mutation. In addition, in the rare case where there wasn't 50 mils of peritoneal fluid, we were even able to get cell-free DNA on peritoneal lavage. We were able to prove that on 20 mils of peritoneal fluid, we could do p53 mutation to confirm it was tumoral. We could find BRCA mutations in 24% of these samples, and we even found a BRCA mutation in the peritoneal fluid in a patient whose FFPE sample didn't identify a BRCA mutation. Finally, we were able to prove that on 100% of our peritoneal samples, we were able to perform genomic instability. So I think this is the first study showing that we can actually use 20 mils of peritoneal fluid to perform a complete HRD testing. The DNA quantity is impressive, the quality is amazing. And that means we can keep precious FFPE samples aside so that in case in the future we need to do biomarker testing, we still have FFPE samples left. We actually think that this could potentially change the diagnostic pathway for women with newly diagnosed advanced ovarian cancer. Well, I just want to thank you, Dr. Lorusso, for a great discussion today. ESGO was really exciting with a lot of new data. And I want to remind the listeners that the slides associated with discussion on key new data presented at the 2023 ESGO Congress are now available on the CCO website. I hope our listeners have found this discussion educational and entertaining, and that this can help inform their clinical practice. Thank you for joining us. Goodbye.
Thank you very much, Dr. Leary and Dr. Lorusso, and thanks to you, the listeners, for joining us. As a reminder, to view the full program, key findings and expert recommendations in gynecologic malignancies, independent 2023 Conference Congress coverage, and to download a highlights slide set including the various studies associated with this discussion, please click on the link in the show notes. As always, thanks for listening.